Hmm. Hello and welcome to this evening's Broad at 15 lecture. My name is Tom Ulrich, and I'm the Associate Director for Science Community, this 8th and final talk in the Broad at 15 series, revolutionary changes in biology and medicine that have taken place since the Broad Institute was founded in 2004. And they've also helped celebrate the parts that we as an institute have played. Our speaker this evening is computational and systems biologist Aviv Regev. Aviv is a core institute member and chair of faculty at the Broad, and director of the Institute's Cell Circuits program and Klarman Cell Observatory. She is also a professor of biology at MIT and a Howard Hughes Medical Institute investigator. Aviv will discuss how recent breakthroughs in single-cell genomics have fueled efforts to build a Google map of every cell in the body, and how such maps are being used to shed light on rare and common diseases. If you have any questions for Aviv, please ask them using YouTube chat, and we'll do our best to discuss them during the Q&A portion of the evening. And if you're tweeting, please use the hashtag #BroadAt15. Without further ado, Aviv, over to you.
Thank you very much, Tom, and thank you everyone for joining this presentation and for coming to listen to our work here at the Broad on the Human Cell Atlas. I'm also going to start with my disclosures. So I'm going to talk today about cells. Cells are the basic unit of life, and each of us is made of about 37 trillion cells, which come in many different types, like neurons, immune cells, muscle cells, fat cells, and so on. At first glance, you may think that every cell in your body is the same. After all, they have the exact same set of instructions in their DNA. But in fact, cells have different structures, locations, and they perform different functions in our body. How come? Well, even though every cell in your body has the same set of instructions, which part of these instructions they will use and to what extent they'll do it, that means how much RNA and protein will be produced based on each gene in our genome, that depends on what the cell needs. So imagine that in the genome, there's just 4 genes, A, B, C, and D. And there are just 3 kinds of cells in the body, neurons, immune cells, and muscle cells. These cells need to do different things. Neurons process signals, immune cells respond to pathogens, muscle cells contract. To do this, they need different proteins that are coded by different genes. So for example, cell 1 may need some of gene A, less of gene C, and not at all of B or D. And so it's going to express multiple copies from A and fewer from C and none from B or D. And I'm going to label this with colors. The darker the blue, the more the gene is being used or expressed. Now imagine that cell C needs each of A and B and C, but none of D. And so this is going to be colored by these lighter blues. And finally, cell 3 might express high levels of B and D. We call this pattern of how much each gene is expressed an expression profile. And you can think of this profile as the calling card of the cell. Because if we know what the cell uses, then we know what the cell is. Cells that are similar should have similar calling cards. Neurons all express similar genes. Immune cells express other genes, and they're all similar to each other, and so on. Now, why does all of this matter? The fact that all cells in our body have the same instructions, but they use different parts of them, it has critical implications when we start thinking about the genes that cause disease. And so imagine that there's a genetic variant that confers the risk for disease. All cells in our body are going to carry this variant, but the disease is only going to manifest in those cell types that use or express this gene. And so knowing our cells, it's a critical intermediary. It's essential for our understanding of how the genes that cause disease act. This is all wonderful. There's only one problem. And the problem is that we don't really know our cells. We don't know how many different kinds of cells there are, where they are precisely in the body, and so on. If you open Wikipedia, it might tell you that there's maybe 300 different major types of cells, and that's across 37.2 trillion cells in the body. But if you ask a neuroscientist, they will tell you that just in your retina, there are more than 100 different subtypes of neurons, and each of them does something different. And so what we need is a map or an atlas that has clear coordinates that would let us know all of our cells and what they do. But now the question is, what would be the coordinates of this map? So I'm going to argue today that the coordinates will be the level of expression of each gene. So let me think with you about a map like this. So imagine there's only 3 genes in the genome, and I'm going to represent each of them by one of these axes, like X, Y, and Z axis. And so, for example, this big cell expresses a lot of gene 3, so it's going to be high on that axis, but very low levels of gene 1 and 2. And so it's going to assume this specific position in 3-dimensional space. Now, our genome does not have only 3 genes. It actually has about 20,000 genes. So there's 20,000 axes or coordinates in this map. And I can't show you 20,000 axes. But math and machine learning algorithms, they can handle spaces like these. They can see the map for us, and they can discover its patterns, as I will show you. So this is all wonderful, but how would we get information to position the cells in the map? Well, for a long time, we actually couldn't do it very precisely or at very high resolution until new lab methods came along that are called single-cell genomics. So in order to explain what that is, let me take you one step backwards to what we did before we had single-cell genomics. Until recently, if we want to measure how much each gene in the genome is expressed, we had to take a piece of tissue that might have hundreds of thousands or millions or even billions of cells, and we basically measured their average. So if you think about each cell as a piece of fruit, we were generating the lab equivalent of a fruit smoothie. Now, if all of your tissue is made just of one kind of cell, just of strawberries, that might not be so bad. But if once in a while there's a special kind of cell, in the fruit salad, 1 in 1,000 is a blueberry, when you make this average, you wouldn't be able to distinguish that it was there at all. In the last few years, there's been major breakthroughs. And in single-cell genomics, we now measure the expression of genes in each cell separately. So you can think of this not as a fruit smoothie anymore, but as a fruit salad. And what is special is that now we can see the different kinds of fruits. We can see how many we have of each kind. And you can even tell the differences within them. For example, not all of the strawberries are the same. You can have ripe strawberries. You can have very mushy strawberries. You can have strawberries that are still very hard. You can have big ones and small ones, darker red ones and lighter red ones. All of these distinctions are now apparent. And at the very latest technological advances are in spatial technologies that allow us to do these measurements when the cells are actually organized together in a tissue. And we can think of this actually as a fruit tart. Today, I'm mostly going to focus on single-cell genomics. And in particular, I'm going to focus on a technique called single-cell RNA-seq. In this technique, we start with a piece of tissue, which we dissociate to make into a suspension of single cells. We then capture each cell individually and tag and isolate its RNA molecules. We can then sequence the RNA, and that's going to tell us 2 things. One is the identity of genes that the RNA came from, and 2, how many copies of them were expressed. Now, because there's so many cells in the human body and so many cells that we need to look at, we need to be able to do this very quickly and very cheaply for very large numbers of cells. One approach that allows us to do this is shown in this movie, which is a result of a technique called drop-seq that we developed together with Steve McCarroll's lab and David Weitz's lab here at the Broad and at Harvard. In this technique, the cells get captured in little droplets in tiny devices. Using this and other massively parallel methods, we can now profile tens of thousands and even millions of cells just in one day. This led to an explosion in the number of cells that we can look at. These are the numbers from the Klarman Cell Observatory here at the Broad. In 2011, 2012, we profiled our first experiment, which consisted of 18 immune cells. But by the end of 2019, we already profiled over 30 million cells at the KCO. But the question is, of course, not just how many cells have we profiled, but what do we learn from them? And what I'll show you is that we learned from these profiles that cells can tell us what we're made of, how we develop, and where disease is. And I'm going to illustrate this to you now with 4 brief stories. The first story is about discovering cells in our body that we didn't even know existed, and that those cells can be important in disease. And so this story starts with the trachea, which is part of the airways, and actually at first with mouse, not human. To know which cells make the lining of the airway, together with Jay Rajagopal's lab, we profiled the RNA of 40,000 cells with those droplets that you just saw. In this plot, every dot is a cell. I cannot show you the cells in 20,000 dimensions, but our algorithm took this information and put it in 2 dimensions, so that if cells were close to each other in 20,000 dimensional space, they are very likely to remain close to each other as points in this 2-dimensional space. Then another algorithm also looked at the data, and it looked for groups of cells that were close to each other. Because if cells are kind of the same kind, they will likely have the same calling card, will express the same genes, and will end up close to each other in high-dimensional space. And so the algorithm will find those, and it's going to color the cells that belong to each such group or cluster with one color, and we will consider those to be one type of cell. And in fact, we found in this way each of the 6 types of cells that we expect to find in the airways, in the trachea. Except that in many cases, what we thought was just one type of cell was actually 2 or 3 different types or subtypes. But then finally, there was this tiny speckle that you see here in purple that did not look like anything that we've seen before. So we asked ourselves, what might that be? So to figure out what these cells might be, we went back to their calling card to find which genes might be uniquely expressed by these purple cells, but not by any of the other cells, and maybe characterize what these cells are. Based on 3 of these genes, we realized that they're similar to cells that were only seen before in frog and in fish. And those cells were called their ionocytes. So we decided to call them the tracheal ionocytes. But also we had a major surprise, because among these genes in the calling card, we found that the cells also express a gene called CFTR, also known as the cystic fibrosis gene, which scientists have thought for the past 30 years or so are actually expressed in a different kind of cell, the ciliated cell. So there's a cell seen in the red dots, which is a very common kind of cell, rather than a rare cell like the ionocyte. But now we found out that it is in fact used by ionocytes and not in the ciliated cells at all. Now, because we first found this in mouse, we then went and we looked at hundreds of thousands of cells from human lungs. And again, we found that it is the ionocyte that expresses the CF gene, the CFTR, and not the ciliated cells. So the next story, brief story that I will tell you, is about asking how did the cells in our body actually come to be. So we all start actually from just one cell. That's kind of a remarkable thing to behold, the fertilized egg. And yet we become many trillions of cells of many different types. This process is called differentiation. And again, I'm going to start with an animal, in this case, the zebrafish. Fish embryos develop very, very quickly. And by 12 hours, there are dozens of different cell types that are organized in tissues. What we want to know is where do these cells at 12 hours actually come from? And here we use another algorithm to actually order the cells precisely and recover back the gradual process that finds the tree of all of the possible paths that the cells might have assumed in order to become what they are, as they diversify and as they differentiate. Now, we can do this not just for zebrafish and not just for early development, but also for humans. So here is the case of human blood cell formation. And here's what an algorithm does. It takes all of the cells, hundreds of thousands of them, which are at first all grouped together in this central position. You cannot really see each of them individually because all of these black dots are really sitting on top of each other. And the algorithm is going to try and find order in these cells. So we're actually following in the footsteps now of this algorithm. And as the algorithm does that, we can find where different kinds of cells are sitting. So here are the stem cells that lead to red blood cells, myeloid cells, which are another kind of immune cell. We will soon see the differentiation of B cells. And we also can find different kinds of T cells and NK cells. So all different kinds of cells in our blood system and how they develop recovered just from these cellular profiles. The third story that I'm going to tell you is about the programs that the cells use. because even when the cells are all of the same type, they shift some of the genes that they use in order to fit the circumstances that they need to adapt for. And this is especially important in diseases when things can start going awry. So I'm going to illustrate it with an example from a cancer, specifically from melanoma. And you might have heard about the remarkable advances of immunotherapies in melanoma through the development, especially of checkpoint inhibitors. So just as a brief explanation, you have T cells normally in your body that identify and kill cancer cells. And that's one of the main reasons that most of the time cancer actually does not develop. It starts arising, but the T cells come in, recognize the mutated cell, kill it, and we go back on our merry way. However, in some cases, the cancer cells can hijack natural mechanisms that the body has in order to shut off the immune system. And in this case, they inhibit and they exhaust the T cells, such that the T cells can no longer come and kill them, and the tumor can develop. The major breakthrough of immunotherapy was to develop what is known as checkpoint inhibitors. These checkpoint inhibitors cut off the ability of the cancer cell to inhibit the immune cell. The T cells are unleashed again, and they can come back and kill the cancer cells. And that brings great benefit to patients. Except that only some patients benefit from immunotherapy, some patients do not. And even those who originally benefit from immunotherapy sometimes develop resistance. And we wanted to understand the basis of this resistance. And so now we can ask, what might be the difference between cancer cells from tumors of patients where there was resistance to immunotherapy versus cancer cells where the tumors were not resistant and and the patient was benefiting from checkpoint inhibitors? From one perspective, these are all cancer cells, but somehow they're different from each other. And because we don't know in what way they're different from each other, the best thing that we can do is just look. And so for this, we decided to look at cells, not from healthy tissue, but from tumors. And now we're comparing patients that were not treated at all to cells from patients that have been treated by immunotherapy but failed. Again, all of the dots in these plots are cells. In the first plot on the top, the cells are simply colored by the patient that they came from. And you can see that the cells are separating from each other based on the patient. This is because tumors are really different from each other. And so the cells of each patient in some way are different from the cells of all other patients. In the bottom plot, I colored them by the treatment. The cells in the black are the cells that came from patients whose tumors did not respond to immunotherapy, and the cells in red are from patients that were not yet treated. But what is really exciting is when we color these cells by a new program that we found that we call a resistance program. This is generally a program that is highly active in those cells, in the black cells that came from patients after immunotherapy. But if you look closely at some of the cells from patients that have not yet been treated, you can find that some of them already express this resistance program. And so this might suggest that patients with cells like these might have tumors that are unfortunately resistant to immunotherapy and might not benefit from it. And indeed, so these might be untreated, such untreated patients with resistant cells might be at risk of not benefiting from therapy. And so when we look at this program, we find that it's present in tumor cells before therapy, and that patients are indeed less likely to respond to immunotherapy if such a program is present. And so now we can ask, is there a way to reverse this program? And by looking at the effect of different drugs on these cells, we found a family of drugs that when given to the cells that have the resistance program, reverts them to the sensitive state. So now we can propose a new option for treatment. Combine the immunotherapy with this drug called abemaciclib. The drug would convert the resistant cells to the sensitive state, and then the immunotherapy will allow the T cells to come in and kill the sensitive cells. We've already shown that this combination is successful in animal models of melanoma, and a phase 1 clinical trial in patients is in the plan. The fourth story that I will tell you is actually on a common disease that has a genetic basis, which is called ulcerative colitis, which is a form of inflammatory bowel disease. Now, human genetic studies of inflammatory bowel disease found many new genetic changes that are causes of disease risk, and in principle could highlight areas to develop new drugs. Now, inflammatory bowel disease and ulcerative colitis, they are not like cystic fibrosis. The disease is not just about one gene that is mutated. It is about many variants that are actually common in many people, including many people who do not have ulcerative colitis. And there are hundreds of such risk genes. And what is difficult for us is that for the majority of these risk genes, we do not really know what they do, and we do not even know in which cell these genes are expressed. And so this opens the question of where should we go? And so to answer this, just like in the airways with the cystic fibrosis gene, we turn to make a map of the cells in the healthy and ulcerative colitis colon, which we did in collaboration with Alex Chalek and Ramnik at Xavier's lab here at the Broad. Now, just like before, each dot is a cell. The cells are colored by their different types in the way that the algorithm found them. The advanced series that we're not looking just at 2 dimensions, we're looking at 3 dimensions, and the cells are organized on these beautiful spheres for us to behold. There are over 50 different types and subtypes of cells that are organized here by our algorithm. Now we can compare these cells between healthy and ulcerative colitis samples. Some of these cells are present only in the diseased tissue, and we do not see them at all, or we see very few of them in healthy patients. These are cells, for example, like these M-like cells, the inflammatory fibroblasts, and specific kinds of T cells. These cells also form new connections in the disease, and they might break down the normal ways in which the different cells in the normal tissue regulate each other and keep each other in check, and that might lead to this uncontrolled inflammation and tissue damage that we see in patients. Now we can ask, how does this relate to the risk disease genes that we find through human genetics? So we can ask for each gene related to the risk of ulcerative colitis, which I'm showing here in the rows, which cell expresses it, which I'm showing here in the columns. Here, the color code is a little different. Red means high expression, blue means low expression. And you can see that in most of the rows, I'm seeing just one red dot. This means that most of the risk genes are expressed by only one or very few specific types of cells. Now, even though there are many hundreds of genes and many dozens of cells, the genes end up grouping together so that they go up and down together within specific cell types based on these programs that the cells need in order to change their states and behaviors. So, for example, a rare cell, like those M-like cells, which really we only see in any meaningful frequency in diseased tissue, has 4 such coordinated risk genes that might all be working together. And now each such group of genes in a specific cell type can become a target for therapeutics. These kinds of maps are useful not just for ulcerative colitis, but they can be used to understand many different genes from many different diseases, all of which might impinge on the gastrointestinal system. So this includes rare diseases like Hirschsprung syndrome, and neurodevelopment and neurodegenerative diseases like autism and Parkinson's that have comorbidities in our gut. And so I showed you 4 stories of how mapping cells can give insight into rare disease, regenerative medicine and development, cancer, and a common disease like IBD. And there are many, many other stories that I could have told you, both from work here at the Broad and from other groups around the world. And so if having a map is so much better than not having one, if it's so incredibly useful, why not make a map of all human cells? And that led us to start the Human Cell Atlas Initiative, whose mission is to create a comprehensive reference map of the types and properties of all human cells, the fundamental unit of life, as a basis for understanding, diagnosing, monitoring, and treating health and disease, just like I showed you in my 4 vignettes. We also fondly call it a periodic table of our cells, thinking about it not just as a catalog of our cells, but also as a way of understanding the principles that give rise to them, and maybe one day being able to predict which cells are there that we have not yet even seen and how they might behave. But I should also point out that humans and their cells are diverse. The scientists that study them are diverse. The places that we live around the world are diverse, and while our genome might stay exactly the same in all geographies, if you go on a plane, which we cannot do as easily these days, and go somewhere else, your cells actually change. They respond to their environment. And so we argued that building an atlas should not be something that's just one lab or one institute or even one country builds alone. And so the International Human Cell Atlas Initiative is a rapidly growing community. It is open to any and all who adhere to its mission and to its values. And to date, we have over 1,800 members from more than 70 countries and more than 1,000 institutes all across the globe, honestly, in all continents except for Antarctica. And this initiative started in late 2016. That's when we kicked off our planning process. Data collection really launched in October 2017, so we're about 2.5 years in. And together, we're really mapping the whole human body, although in our first effort, we have a particular effort on 14 organs and systems, and they include things like the nervous system, the gut, the lungs, the liver, the reproductive systems, the immune systems, and more. We've already collected millions of cells from hundreds of individuals around the world. These are already leading to valuable insights now that we can start querying this emerging atlas. And this really leads me to the last example that I want to share with you today. And I'm going to illustrate how valuable the atlas has already become by showing you how the Human Cell Atlas community queried the atlas for the cells which are the likely targets of SARS-CoV-2 or the COVID-19 virus, which is, of course, front and center in all of our minds. And so just as a brief introduction, for SARS-CoV-2 to infect cells, it needs to bind a receptor on the cells, on our cell surface, known as ACE2, that's a human gene. And in order to really productively get into the cell, it also needs the human cell to provide it with an additional protein known as a protease. And there's at least 2 of them, and probably more, that can fulfill this function, one called TMPRSS2 and the other called cathepsin L or CTSL. There's probably additional ones of these proteases. But knowing that this is the basic code that the virus needs for entry, this is like the key that it needs in order to unlock the door into human cells, one can now ask oneself which cells might be infected by the virus simply by virtue of expressing these needed entry genes. And given a big atlas of many cells across the human body, we can start querying the atlas for cells across the healthy body that express ACE2 and one of these proteases. And this is what our international community decided to do across the Human Cell Atlas through the biological network for the Human Cell Atlas known as the Lung Biological Network. And I want to emphasize here that, again, this is not something that one group could have done alone, because the diversity of tissues we wanted to study and the number of individuals and samples we needed in order to just look for cells in the human body required data and expertise from groups all across the globe, including from the Broad Institute. And so this effort has already led to multiple key insights that span the cells that are likely first infected and involved in transmission, some of the immunological responses in which those cells might be involved, and also knowledge about the relevance of different types of model organisms, like non-human primates and mice that we might want to use in order to do further studies and identify therapies, as well as what might explain the differences in pathogenesis between individuals of different ages, the impact on different organs in the body, and the mechanisms that might be employed. And I'm going to give you a little bit of a glimpse into some of these studies in the last part of this talk. And so the first step that we did was to query the atlas across 25 different tissues in the most agnostic way possible, asking for what might be possible target cells. And we found those cells in a lot of places. So we found them in the nose and in the lung and in the airways, which are places that you might expect to find cells infected by the virus, since we all know the symptoms now of this respiratory virus. But we also found many such cells in the intestines. We found cells in the liver, in the eyes, in the heart, in the pancreas, in the bladder, in the testis, in the kidney, in the brain, and also in the olfactory epithelium, which is the sensory organ that gives us the sense of smell. Now, some of these tissues, for example, the eye and the intestines, can be involved in transmission, and that's incredibly important for us to know. Others of these organs were already implicated in the pathology of COVID-19 disease. For example, patients die from heart disease. There's kidney failure. There's the lack of sense of smell in the olfactory epithelium, and there's also neurological symptoms in the brain. But what we identify is not just the fact that these organs are implicated, we find which cells in these organs are implicated. And this is important for two reasons. In many of these cases, these cells are not so prevalent. And when they're not so prevalent, if you had just looked at the organ itself and you didn't look at the individual cells, you actually would never have implicated that organ. The second reason is that once we find the specific cells, we can start understanding the way in which the virus might be interacting with its host. And so let's take a little bit of a look at those cells as well. For example, we found that muscle and blood vessel cells in the heart might be infected by the virus, and this might explain myocarditis in patients. We found these cells in the sensory organ of smell, and as I said, the patients lose the sense of smell. We even found cells in the brain, and there's now increasing evidence of neurological symptoms and inflammation in the brain that might be affecting patients. Okay. Next, we asked, is there something about the cells, especially in the lung and airways, which are the main site of infection and disease in most patients, that might explain some of the phenomena associated with the disease? For example, you might all know by now that this disease seems to afflict much more the elderly than it afflicts the young, and in particular, the children seem to have very few symptoms, if at all, and very rarely any severe disease. There's also some evidence that men might be affected more than women, and there's an open question on whether smoking does or does not impact the course of disease. And so for this, our network assembled data from 22 studies across the Human Cell Atlas that spanned 164 donors, analyzing 282 samples from the nose, the airways, and the lung, to a total of 1.17 million cells. And this number of samples was needed in order to make statistically robust associations. And as we made them, we found that the level of the viral receptor, that gene, ACE2, is higher in specific cells in the lung as individuals age, and it is also higher in men than it is in women. Smoking is more complicated. It is increased in some cells in the airways, and it decreases in others. You can also focus on specific phenomena, and some of these patterns might explain, some of these differences that you see between individuals. For example, you can focus on some phenomena. Young children seem to very rarely develop severe disease. And indeed, the viral receptor is extremely low in cells from pediatric samples, suggesting that especially in the lungs, there are far fewer opportunities for the virus to infect. Finally, we can ask, is there something unique about the gene programs that cells with the receptor use compared to the same kind of cells, but they do not express the receptor? So we're not comparing different kinds of cells, we're asking within a particular kind of cell. Just like the kind of question that we asked about the cancer cells of immunotherapy resistant and sensitive patients. And we indeed found that these cells, even in the healthy body, have an immune-like program. And you might have heard that patients who develop COVID-19, severe disease, often have strong immunological symptoms and responses. So this opens up an hypothesis that, of course, still needs to be studied, that it is possible that this program might contribute, if the cells are infected, to this uncontrolled immune response. And so, by querying the atlas, we learned about the cells that may be involved in infection and transmission. We learned about the cells that might be involved in pathogenesis, both in mild disease and in severe disease, such as implication in the heart, in the brain, in the kidney, and more. We learned how these cells might be different across the ages, that they might be different in males versus the females, in children, and in smokers. And we learned some of the programs that these cells use in the healthy body. I didn't show you, but in additional studies, for example, we also learned about in which way model organisms can be beneficial because they're similar to the human, and in which ways they are actually quite distinct because they are dissimilar, and we shouldn't later on be fooled by their results. Now, reaching these and many other conclusions in these studies requires concerted international efforts, which now continue as the Human Cell Atlas here at the Broad and across the world begins to study COVID-19 patient samples, as well as we study samples from healthy individuals and from many other diseases. These are only the early days for the Human Cell Atlas, but we're now well on our way to hit our first draft at the 5-year mark, and to work on a comprehensive atlas of 10 billion cells in our decade-long effort, as we aim to build a comprehensive reference map of the human body. I thank you very much, and I'll be happy to take questions.
Great. Thank you very much, Aviv. That was a fantastic talk. Just a reminder to the audience to please, if you have any questions, to add them to the YouTube chat. And we have had a few come in if you're ready. So here's one that has come through. Does the process of collecting cells for single cell sequencing affect the data you generate? Does it affect the cells in some way that would change their expression profiles?
This is an excellent question. So the answer is kind of a yes and a no, and I'll elaborate a little bit. So in many studies of single cell genomics, you have to start with fresh tissue, and you are right that if you wait, things happen, cells respond, and so you need to act extremely rapidly. This has actually been inadvertently one of the joys of our field, that it brought together many communities, surgeons and pathologists and molecular biologists and computer scientists to really work hand in hand to solve this problem. And for us here at the Broad, the integrated community spanning the hospitals, MIT, Harvard, and the Broad Institute is what allowed us to pioneer many of these studies of patient samples. At the same time, over time, we identified additional techniques that allow us to stop the clock sooner. So one great example of that is what we call single nucleus RNA-seq, when we no longer analyze dissociated cells, but we actually analyze nuclei and often RNA that's attached to the nuclei from the outside. Nuclei can be isolated from lightly fixed and also from frozen tissue, and you can also isolate them from tissue that otherwise would be very, very difficult to dissociate to individual cells, for example, from aged brain. And as we started doing more and more of these single nucleus analyses, that allowed us to both target tissues that otherwise would be difficult for us to study, look more and more at patient samples, and also stop the clock immediately with freezing of tissue, and as a result, avoid these changes that would otherwise sometimes happen in cells.
Oh, great, thanks, Aviv. And actually a somewhat related question that's also come in. If you were to take stem cell derived cells or cultured cell lines from different cell types that are within the same organ in nature and run single cell sequencing on those, do you think you would get results that are similar to what you would get from the original tissue itself?
That's a fantastic question. We call it adjudication, meaning taking a model system, in this case an organoid or stem cells that you grow in culture, and comparing it to the cells in the human body. And the Human Cell Atlas really, and these techniques really allow us to do it. And the answer is, sometimes it depends. And I'll give you an example from gut organoids, which is one of the greatest successes of the stem cell biology. You take stem cells, intestinal stem cells in this case, and you can grow an organoid that has a lot of the epithelial cells in the gut. And by and large, for most of the kinds of cells that we develop, you get a very good mimic of the cells of the gut, of the epithelial cells of the gut. But there are two kinds of cells called planet cells and goblet cells that normally would be two distinct types of cells, and in an organoid, they don't fully separate from each other. They kind of remain sort of a chimera or a hybrid or a mishmash of the two different kinds. There are cases where we try to basically use what we know already as what looks like, is how the normal body cell looks like as our target actually to screen towards that direction. And the combination actually of two favorite technologies, single cell RNA-seq and CRISPR, allows you to develop screens of this kind as well.
Great. Another question that's come through, are there organs or cell types that are really easy to study through single cell sequencing and ones that are really hard to study in this way?
Excellent question. I would say immune cells are one of the greatest friends of the single cell biologist, partly because they are naturally supposed to exist as suspensions in the blood, and also they go in and out of tissue easily. Again, it's their natural job. So they tend to profile easier and faster, but they can be more susceptible to change, meaning they can be more responsive to the environment. There are cells that are definitely harder. Neurons, especially adult neurons and neurons in aged tissue, are incredibly difficult to dissociate, and honestly, the field has moved to analyze their nuclei mostly. Some of the epithelial and stromal cells can be quite sensitive and die during the process of dissociation. So I would say that the spectrum is roughly so. Neurons, adipocytes, fat cells, and muscle cells are difficult to impossible to dissociate as cells, and we usually study them at the nucleus level, or now at the tissue level using spatial techniques, whereas immune cells might be on the easier end of the spectrum.
Okay, great. You gave examples of single-cell RNA studies. Do you see how other forms of omic data like genomics, proteomics could contribute both to our better picture that we get out of single-cell sequencing, but also to the Human Cell Atlas as a project?
Absolutely. And the Human Cell Atlas, as I tried to emphasize in the beginning, is not just about single-cell RNA-seq. It's about multiple single-cell approaches, their combinations, which is what we call multi-omics, and their spatial organization. It's just in this talk, I focused on one aspect. So I'll try and parse it out. In the genomic side, we can get remarkable information about the genetic state of a cell, which is important, obviously, in diseases where you have genetic changes, such as cancer, but also some other diseases where you have somatic changes, as well as when you're thinking about the genetic differences between individuals. Including the DNA side is very important. As you move from the DNA to how the DNA is organized for regulating gene expression, single-cell chromatin profiling, for example, single-cell ATAC-seq, is incredibly helpful at understanding the regulatory mechanism, and also especially tends to target regions, regulatory regions, which are where most of the common genetic variation in our genome actually lies. It's less in coding regions and more in non-coding regions. And then we have the RNA that we already discussed. Also, RNA and chromatin tend to reflect different timescales for cells. RNA is more about the immediate and the current, what the cell is doing now, or it's anticipating and doing right away. The proteins are on a longer timescale, and the chromatin often on an even longer timescale than that. So each of them gives us a different temporal view of the cells. They're complementary to each other. The proteins are also the business end, and there are many changes that happen at the protein level. For example, protein modifications, the location of the protein in the cell, all of which have an impact on their function. So having all of those absolutely improves our situation. The conceptual framework is the same. You can think of it as having even more axes than we had before, or more of these high-dimensional spaces that machine learning now increasingly aligns for us in order to relate things through these different lenses. I will also want to give a plug to what we call multi-omics, which is when you measure multiple measures at once. If you know the chromatin and the RNA simultaneously, you can understand how gene regulation works better. If you have genetic information, not just about the nuclear genome, but for example, about the mitochondrial genome, we have now learned that this gives us lineage information that allows us to trace the history of cells as mutations accrue in them and relate it to the state of the cell. That opens up a lot of beautiful questions in developmental biology that otherwise are inaccessible for us in humans. And then, absolutely critically, those fruit tarts, which are a major aspect of our work now, is to not just think about the cells as they're, dissociated from each other, but at the cells in the context of the tissue next to other cells. Tell me who your neighbor is, and I'll tell you who you are. And how cells form together these communities and modules, those are the functional units of tissues, and to a large extent, the functional units that go badly in disease. And so spatial ways to identify which cells are sitting next to each other are also remarkably helpful for us. All of these are part of the Human Cell Atlas.
Okay. And as the Cell Atlas has come along, roughly how long does it take to build what you would consider to be a good map of an organ?
Yeah, that's a great question. I think for a census of cells now, you can work within the span. Today, when we started, this was not the case, but today, within the span of a year or less, you can collect the information. The assembly of the map is harder. It's a computational challenge. But I would say that for many of the organs probably today, with concerted effort, just a census of the cells that's there, which types are there, you can get quite rapidly. The challenge is several-fold. the organization histologically of the cells, so the spatial map at the histological level, the organization anatomically, not just knowing which cells are there, but where they are in different parts of the organ. These things take a longer amount of time, and also understanding the inter-individual variation. To a first level of approximation, cells from different individuals actually are really similar. But at the second level of approximation, of course, there are differences between them. And we want to understand these variations and differences, and that expands the scale of the map. Our sense is that within the first 5 years, we will be held to what we promised of our first draft, and maybe we're even a little bit ahead of schedule on it, although time will tell.
All right. And then when you find a brand new cell population within your data, how do you tell what type of cell it is? How do you characterize it?
That's a fantastic question. I'll start with the example of the ionocyte, but I'll also try to put it in context. So the ionocyte is a great example because it's such an outlier on the one hand. So it wasn't really similar to anything seen before because it wasn't seen in that form before. But it was seen in another species. So it's very similar to how do you figure out what a gene does? One of the best ways to figure it out is looking where it is else in another organism and saying, but I know something there. That's what gave it its name, the ionocyte, and that was because the gene program that it uses is actually conserved. And we increasingly know, especially from studies of human and mouse and now additional organisms, that there is a decent level of conservation in many of these cases. So it allows us to transfer information that we have from one area of study to another. The other way in which we can guess our way around the cell is thinking about how it relates to other cells in this high-dimensional space. So often it will have cells that it is closer to, and you could say, oh, so it comes from that family. I might not know its direct role, but I know that it's within this kind of class. So there's similarity to other species, there's relationship to other kinds, and then there are the genes and the gene programs that the cell uses, and often where the cell is in the tissue, which structures it forms inside. Now that you know its calling card, you can use that in order to probe tissue and see where it is. But this does not mean that we immediately know. We have to convince ourselves first that the thing is real, that it reproduces, that if I looked at it through another lens, if I didn't use RNA and I used something else, would I still find the same grouping of things? The more we see that, the more we believe that it is a new entity. And then finally, it's very hard to replace functional experiments where you're starting to manipulate that system and trying to see what happens to it. These are, of course, not doable in humans, but they're doable in models, including models in a dish and animal models.
Okay. We had a couple of coronavirus questions that have come in.
Yes.
No surprise. So has anyone looked at ACE2 receptor levels and to see if they're different in people who are taking ACE inhibitors or similar drugs?
This is like an invited question. It's a slide I didn't show and almost showed. So my wonderful colleague here at the Broad Institute, Patrick Elinor, is part of the Precision Cardiology Lab. Patrick is a cardiologist, and he heads an effort really focused on the cardiovascular system and the heart, and collected a beautiful atlas preceding COVID-19 of both the healthy heart and of patients with cardiovascular disease, some of whom were on ACE inhibitors. And so he actually has a study on medRxiv, also in collaboration with the Human Cell Atlas Lung Biological Network, expanded beyond the lung by now, that actually shows the impact of ACE inhibitors and also cardiovascular disease. I don't want to belabor the details, but I will emphasize that, in fact, there is an impact both in cardiomyocytes, in the muscle cells of the heart, and in pericytes and fibroblasts, so more connective tissue and vascular cells. The impact is opposite. And so that might have led to some of the confusing, it was very confusing to reason through what might be happening in the heart. When you average both of these in the bulk, you kind of lose the signal. So one goes up, the other goes down. It seems like nothing is happening. When you look at the single cell level, there's actually something happening there. Now, the study would have to be expanded to a much larger number of individuals. And I also want to emphasize this. These are all studies that precede COVID-19 itself as a disease. And once individuals are infected, things can change dramatically in their body. Of course, we actually assume things change dramatically in their body. Now, in our efforts, we are starting to profile tissues that come from COVID-19 patients, which is incredibly sad, but that we hope will actually help understanding some of these questions.
All right. And then one last question on the theme of change that you just touched on. Is it common for the expression of receptors like ACE2 to change with age?
I do not know this as a general answer, but it is not uncommon for the expression of genes to change with age. In another study, there was a finding that showed from groups that collaborate again with the Human Cell Atlas from UCSD, part of an effort called LungMap. They show that the change of ACE2 expression with age might have to do with overall reprogramming of the cell's chromatin and in these specific lung cells, with the reprogramming of the cell's chromatin. And I should point out that some of that involves likely inflammatory transcription factors or antiviral transcription factors like IRFs and STATs for the aficionados. And so there might be an exposure aspect, for example, over age that over time leads to this change. ACE2 is one of the genes with extremely complex regulatory mechanisms and patterns. It's expressed in many tissues, likely regulated in many ways, and which we should be careful not to over-speculate.
Fantastic answer, fantastic talk, and thank you so much for joining us this evening, Aviv. And thank you also to everyone on YouTube who's been joining us tonight for the talk as a whole. We hope you enjoyed tonight's talk, and if you like what you heard, then consider watching all the other talks from the Broad series. You can find them all on our website at broadinstitute.org/15. And you can also be part of the conversation on social media by using the hashtag BroadAt15. To find out about other live-streamed events and new videos from the Broad Institute, subscribe to our YouTube channel and tell your friends and family to subscribe as well. Thank you so much for joining this evening. We hope you enjoyed it, and we wish you all a good night.